And so I am really thrilled to introduce today's speaker, Mario Penso. He's a rising star in the neuroscience community, and we're really excited that he's here today to share some of his work. So Dr. Penso received his BS from the University of Puerto Rico, where he conducted research on the neuroprotective effects of GABA receptors and nicotinic receptor activation. He went on to receive his PhD in neuroscience from Albert Einstein College of Medicine under the mentorship of Jose Peña. And during graduate school, he carried out some really beautiful studies in one of my favorite systems, the chick embryo, in the inferior colliculus, where he discovered several novel forms of synaptic plasticity and related retrograde signaling to modulate synaptic events. So for his postdoc, he moved to Cold Spring Harbor, where he continued working in the field of synaptic plasticity, this time in the rodent amygdala, under the mentorship of Bo Lee. He carried out a series of really fascinating experiments identifying the paraventricular nucleus of the thalamus as a critical presynaptic partner in the regulation of fear learning and memory, and has really become, a leader in this field. In 2015, Dr. Penso opened his own research group at the National Institute of Mental Health, where he directs the unit on neurobiology of affective memory. His group has been doing some really elegant studies dissecting neural circuits involving both emotional and motivational processes, as well as the susceptibility to stress. Recent work includes publications in Nature, Nature Neuroscience, Nature Communications, and Journal of Neuroscience. He's won numerous awards for his research, just a few including the Young Investigator Award from the Brain and Behavior Research Foundation, as well as the Harvey L. Karp Discovery Award from Cold Spring Harbor. And actually, perhaps as impressive as his research accomplishments are his, amazing commitment to mentoring. He's won a ton of awards in this area, including the NIH Graduate Student Council Outstanding Mentor Award, the Special Act Award, and the NIMH Outstanding Mentor Award, which he's actually won 2 times just in his short time at NIMH. I could probably go on for the full hour about all of his accomplishments, but I will yield the floor to our speaker and welcome Mario Penso.
Thank you very much, Ming, for the kind introduction. It's really a pleasure to be here with you today. Today I'm going to talk a little bit about work that my lab has conducted recently, particularly on the role of the paraventricular nucleus of the thalamus in fear-related processes. As being mentioned, this is an area of the brain that I got interested in when I was a postdoctoral fellow at Cold Spring Harbor Laboratory, where we had some interesting findings about the contribution of this area of the brain, and particularly its projection to the central amygdala in regulating fear memory. And over the last few years in my lab, we've been trying to expand on our understanding of how this region of the brain regulates fear-related processes. The focus of my talk today is going to be largely centered on that. Okay. Let's see. All right. So my laboratory, more generally, is interested in understanding the neural mechanisms that lead to emotional state and emotional related processes. And to address this question, we try to undertake on this multidisciplinary approach that ranges all the way from molecular processes to cell physiology, circuits, and ultimately trying to understand how these processes regulate behavior. And as I mentioned, in my opening slide, one of the emotions that we're particularly interested in studying is fear. And of course, we address this question by, or have been addressing this question using animal models, specifically mice. So obviously, when you talk about things like emotion and fear in animal models, that can be, kind of a heated subject, right? Because we tend to think of emotions and particularly fear from the perspective of subjective reports like feelings and whatnot. So one of the things that I like to do when talking about these terms is to try to arrive at a specific definition, and try to define specifically what we mean when we talk about emotion. And I think from this perspective, one particular framework that I found, we found quite useful in our thinking, is that which was proposed by David Anderson and Ralph Adolph, which is this idea that emotions can be considered to be central states that are triggered by stimuli, and the stimuli can be either exteroceptive or interoceptive, and that lead to a series of readouts of this central emotional state. And these are readouts that we can measure, right, in a laboratory setting. So when thinking of applying this particular framework to fear, which is the question that we're interested in, we can, of course, in animal models, measure a lot of these readouts of these emotional states, with the exclusion of subjective reports, which is still a category that is reserved for human subjects. But I'm sure most of you are familiar that in our field, in neuroscience, we've come to learn a lot about neural processes associated with fear, particularly by focusing on observed behavior in rodents in particular. And this has been via the use of mainly Pavlovian classical conditioning paradigm, which I'm going to describe a little bit more in the next slide. So these classical paradigms, which you guys are all familiar with, involving association of a rather neutral stimulus like a tone with a aversive outcome, like in this case a food shock. The use of this paradigm has been very powerful in neuroscience, and has led to this particular amygdala-centric view of fear, largely because early studies identified the lateral portion of the amygdala as a region of the brain where these associative memories are formed, right? And this is linked to synaptic plasticity in this region, so this is the view that has dominated the field for various decades. This idea that the amygdala is like the fear center of the brain, using this task, of course, we've come to learn a lot about the mechanisms that underlie this associative memory formation, but also the expression of a very specific type of defensive response, which is freezing behavior, since this is the main, the dominant observed behavior in studies of Pavlovian conditioning. So naturally, when you think of this, you realize that the use of Pavlovian conditioning is limited in the sense that it doesn't yield as rich of information or any information actually about the brain processes that participate in the orchestration or modulation of other type of defensive responses. One which we're really interested in my lab is avoidance behavior. And much less do we know about the mechanisms that drive the selection between these opposing forms of defensive behaviors. And I think that this is something that's really important to think about, because although we study defensive behaviors in the laboratory settings, in sort of like reductionist approach, in nature, these type of defensive behaviors are displayed as a function of the spatiotemporal distance from threats in the environment. So when threats are highly imminent, right, they're pretty close, animals will engage in behaviors that are more reactive, like freezing behavior or flight and jumping behavior. Whereas like when threats are much more distant, this is what we refer to as the low imminence part of the spectrum, animals still engage in other type of in defensive behaviors, but defensive behavior that are actually more flexible and cognitively regulated, like, for example, risk assessment and avoidance behavior. So I think this is this threat imminence continuum is kind of like a useful way to think about defensive behaviors. So for my talk today, I'm going to focus particularly on this behavior, avoidance behavior. And part of the reason we're interested in this is kind of like nicely summarized here in this one chart from a review article that was published a couple of years ago by Chris Kane, where you can see that while early on there was a lot of interest in understanding active avoidance behavior, over the last couple of decades, studies of fear conditioning actually have dominated the literature, and there's been like kind of like sort of like not so much development in our understanding of active avoidance behavior as it has been for fear conditioning and its associated defensive outcome, which is freezing behavior. So, having said this, this is not to say that we don't know anything about active avoidance behavior. So here I'm going to actually just briefly outline how we think about active avoidance behavior, particularly when using this classical shuttle box task where animals are learn to associate shuttling to the adjacent compartment when they're presented with a warning sign to be associated with the avoidance of a food shock. So the animals are trained in this shuttle box where they learn every time that there's the warning sign, could be a light or a tone, that if they shuttle to the adjacent compartment, they will prevent the onset of a food shock, and they learn to do this really well. And this is the type of active avoidance response that we're measuring. So when people started using this task, people started to think about the processes that are taking place as animals learn the task. And one way that this has been described is as this two-stage process in which early on in training, the initial processes that take place is this Pavlovian association, where the animal is still simply associating this warning sign, this tone, with this aversive outcome, which in our case and most of the time in literature is a food shock. And this is consistent with studies of Pavlovian fear conditioning. This is a process that is largely dependent on amygdala circuit, the lateral amygdala, which is important for the association, and then the central nucleus of the amygdala, which is critical for this freezing behavior. Now, what you can see here is that as with the passage of time, as animals undergo additional training, they start to begin to adopt this goal-directed response, which is in this case the avoidance behavior, and their learning of this response is associated with a decrease in the expression of the Pavlovian response, the freezing behavior. And this is associated with essentially like a remapping of the underlying neural circuitry, where the behavior is less dependent on the central amygdala and becomes actually more dependent on the ventral striatum, which is important for goal-directed behavior. Of course, with the passage of time, behaviors can be, this active avoidance behavior can become habitual, and it is thought that this is the circuitry underlying this behavior resembles a circuitry that has been previously described in the context of habitual appetitive behavior, like the dorsolateral striatum, and there's actually some newer ongoing work in addressing this particular question. Now, one thing that I'd like to bring to your attention here is that you can immediately see that when thinking about transitioning away from these Pavlovian responses to this instrumental active avoidance behavior, it seems like these two regions of the brain, the central amygdala and the nucleus accumbens, are particularly important. And in fact, there's been studies in the past, and I'm only going to highlight some studies that are particularly relevant to my talk today, but there's been many studies that have looked into this. But one study that is that I want to highlight is this study by the group of Joe Ledoux from 2010, where they demonstrated that silencing, or in this case actually lesioning of the central nucleus of the amygdala, attenuated freezing behavior, consistent with the idea that this region of the brain is required for freezing behavior. But this manipulation actually led to increased active avoidance behaviors in animals that had this lesion. In a separate study, the group of Gregory Quirk, they demonstrated that silencing of the ventral striatum led to exactly the opposite outcome, which is that these animals show impairment in active avoidance behavior, but they had an increase in freezing responses to the condition or the warning signal, right? So this data seems to suggest that these two regions of the brain are in this antagonistic relationship for the control of this type of defensive behaviors. We suggested to us that perhaps regions of the brain that communicated directly with the central amygdala or the nucleus accumbens could play an important role in biasing the selection of these different type of defensive behaviors. Now, naturally, the first region that comes to mind when we think about this is the basolateral amygdala, because it's a region of the brain that has been shown to send innervation to both the central amygdala and the nucleus accumbens and participates in these behaviors. Studies over time had shown that the necessity of the BLA for the expression of these type of behaviors seems to decrease over time. So this suggests that perhaps other regions of the brain are equally or perhaps more important in regulating these two type of defensive behaviors and perhaps biasing the selection of these behaviors. And this is what led us to the paraventricular nucleus of the thalamus, an area that I've been interested in the past, particularly because of his strong innervations of limbic structure. And in particular, two of his major output targets are the central amygdala and the nucleus accumbens. And in fact, when I was a postdoctoral fellow at Cold Spring Harbor, we showed that this projection from the PVT to the central amygdala is actually critical for the expression of conditioned freezing behavior. And this is something that was also replicated in rats by Fabrizio Del Monte and Gregory Quirk. But the question is whether the paraventricular nucleus of the thalamus and its projection to the nucleus accumbens play a role in avoidance behavior. And this is the hypothesis that we wanted to test, that actually we think that PVT might be important for avoidance behavior via this projection to the paraventricular, to the nucleus accumbens. And this is a question that a very talented postdoc in my laboratory, Jun Ma, was set to address some years ago when he joined the laboratory. So here I'm just showing a brief outline of what my talk today. So for the first part, I'm going to actually focus on work that was recently published. This first authorship publication by June that was published towards the end of last year, where we find that the paraventricular nucleus of the thalamus seems to shape the selection of avoidance and freezing behavior. And this is what I will mainly focus on today. And then for the second shorter part of my talk, I'm going to tell you about some new exciting observations that we've collected since the publication of this other work that relates to the role of prefrontal cortex input to the paraventricular thalamus in the shaping of this type of defensive behaviors. Okay, so when June started this project, he was pretty intrigued about determining whether neurons in the paraventricular nucleus of the thalamus that project to these targets, the nucleus accumbens and the central amygdala, actually belong to similar or distinct populations of PVT cells. So to address this question, he injected in wild-type mice retrograde tracers that were coupled to a fluorescent dye, so that he could see neurons in the PVT fluorescently labeled that project to either of these two targets, the nucleus accumbens in one color and the central amygdala in another color. So when he sacrificed these animals and he looked at their brains, he found that you can see very nicely here neurons in the PVT that project to the accumbens, and in red neurons that project to the central nucleus of the amygdala. And when he quantified this data, he observed that in general, many more cells in the PVT project to the accumbens compared to the central nucleus of the amygdala, which is actually consistent with previous publications. Interestingly, he observed that the level of collateralization between these two type of PBT cells, central projectors or accumbens projectors, was relatively low. So perhaps about 20% of accumbens projecting cells also project to the central amygdala, but also roughly 30% of cells that project to the central amygdala also project to the nucleus accumbens. Importantly, we find that the vast majority of these neurons that project to either central or amygdala or the nucleus accumbens are positive for or express D2 receptors. This is important because in previous studies we've shown that D2 expressing neurons of the PBT belong to a specific class of PBT neurons that are engaged by aversive stimuli. So it makes sense that perhaps this is, the population of cells that is important for regulating fear behaviors. So after identifying these two, perhaps these parallel streams from the PBT to the accumbens and the central amygdala, we wanted to assess whether these neurons of the PBT signal active avoidance behavior. So to address this question, June used the genetically encoded calcium sensor GCAM6, which he expressed in D2 positive neurons of the PBT of D2 cremi, since I mentioned that most of these projection cells of the posterior PBT are actually labeled by D2. So he expressed GCAM into D2 neurons of the PBT, and then he implanted an optical fiber right above the PBT so that he could collect bulk changes in fluorescence during the behavior. So again, these animals were then trained in the active avoidance tasks that I mentioned briefly during my introduction. Essentially, animals are placed in this shuttle box that is divided by a hurdle, and they're trained such that a tone is played for 15 seconds, and after the tone is played, a shock is delivered until animals shuttle to the adjacent compartment. They typically shuttle pretty quickly when the shock is delivered. And over time, they learn that if they shuttle during the presentation of the tone, this will avert presentation of the shock. So this is the avoidance response. Here, I'm showing you a short clip where you can see what the signal from these neurons looks like during the task. This is an avoidance trial where here you can see that the cue started playing. This is the calcium transient here. And here the animal has about 15 seconds to shuttle or he will get a shock. And you can see that when the animal shuttles, there's this strong response, calcium response in these D2 positive neurons of the PBT. So after, imaging animals that have been trained in this task, June, and by the way, you can see here that the behavior actually resembles pretty much what I showed in my introduction, which is that early on animals are displaying high levels of freezing during the tone presentation, but over time the freezing diminishes and the active avoidance response increases. So after animals were trained in this task, and June imaged them for the last 2 sessions of training, he divided trials between classified trials into either avoidance trials or failure trials. So the avoidance trials are the trials that, I just show you an example of where the animal successfully avoided during the tone presentation. But a failure trial is a trial in which the animal didn't avoid during the tone and instead received a shock, in which they ultimately escape by shuttling across compartments. So this is here you can see the summary data for all the trials that June imaged here, which is essentially ranked by the latency to avoid. And in general, what we see is that shuttling behavior is nicely associated with an increase in calcium signal in these D2 positive neurons of the posterior PBT. In fact, here you can see this black line actually depicts the actual the expected duration of the tone, which is 15 seconds, like I mentioned. And here, this is just the average of the tones from all these trials in which the animals avoided. And you can see that termination of the tone nicely matches with the increasing calcium signal in these D2 positive PBT neurons. And this is something you can also observe in these individual trials in which each successful avoidance trial was associated with this rise in calcium signals from these D2 positive cells. Now, this is all quantified here. You can see that indeed If we look at the area under the curve during the tone presentation, you can see that in general, for avoidance trials, this was associated with a significant increase in activation of these neurons, whereas during the failure trials, we actually see a reduction in fluorescence in these neurons. And we were wondering as to whether this reduction during the failure trials could be associated to behavior, because during these failure trials, the animals spend most of their time actually freezing. And you can actually see that here, that when we quantify the level of freezing, that the time that animals spend freezing across avoidance trials and failure trials, in general, they spend much more time freezing during the failure trial. So we wanted to look at this more closely by looking at the calcium signal, as a function of very specific task-related events. And this is what I'm showing you here in the next slide. And essentially what you can see here is that in general, for both avoidance and failure trials, these D2 positive cells of the PBT show an increasing calcium response during the onset of the conditioning stimulus, so the tone. And similarly, there was the activity of these neurons was associated with a rise during maximal velocity, which we use as a measurement of the vigor of the behaviors as animals shuttle across compartments. So you can see that from the video that I showed you earlier, that this behavior is pretty vigorous and the animals shuttle with a lot of energy. So we look at the maximal velocity as animals shuttling as a measurement of these active avoidance behavior. And you can see that for both actively avoided behaviors, or where animal avoided a foot shock, these responses look very similar in these neurons. In contrast, when the animals engage in freezing behavior, you can see here time 0 is when the freezing episode starts. You can see that freezing is associated with an attenuation of calcium signal. So this data suggests that these D2 positive neurons of the PBT are signaling during avoidance behavior, but that their response is actually attenuated during freezing behavior. So naturally, the next question that we wanted to address was whether the activity of these neurons was actually required for active avoidance behavior. And to address this question, we use optogenetics. So in this case, June expressed the inhibitory opsin halorhodopsin, specifically on D2 expressing neurons of the posterior PBT. And he implanted an optical fiber above the PBT so that he could silence the activity of these neurons at a very specific time by delivering yellow light to the brain. And here you can see that here's what the data looks like. June trained these animals in active avoidance, and after he trained, the animals were fully trained. He subjected them to 3 additional test sessions. And you can see here that in general, these animals display pretty high avoidance rate, both control animals, which are the black line, and halorhodopsin expressing animals, which is the red line. And here, notice here in the data that's normalized to this first test session, that in the halorhodopsin session, in this case, halorhodopsin was stimulated simultaneously with the delivery of each one of the tones. So this is very restricted to tone presentations. You can see here that when you silence these PVT neurons during the tone presentation, this significantly impairs avoidance behavior. And this reduction in avoidance behavior is also associated with an increase in the latency to avoid, which you can see here. And at the same time, notice here that in these animals that display this decrease in avoidance behavior, they also show a significant increase in freezing behavior during the light test session. So this data suggests that not only silencing these neurons is impairing avoidance behavior, but it's actually facilitating freezing behavior in these animals. And this actually is reminiscent of the study that I mentioned during the introduction from Gregory Query's group, where they show that silencing of the ventral striatum with mucimol leads to a very similar outcome, where active avoidance behavior is impaired and freezing behavior is enhanced in these animals. While this was pretty interesting result, and we're actually quite excited by this data, we were also puzzled because in previous studies, we and others have shown that a subset of PBT neurons that project to the central amygdala actually is required for driving freezing behavior in fear conditioning. We were kind of intrigued by the fact that silencing these D2 positive cells actually unmask freezing behavior and attenuate avoidance behavior, and that the activity of these neurons actually decreased during freezing behavior. It's kind of like contradicting perhaps previous finding that we and others have had. So we wanted to look a bit deeper into this, and one way that we thought of it was by imaging the same type of neurons, these D2 positive cells of the PBT, in fear conditioning. So June took a different group of animals, trained them in fear conditioning, and then he imaged the activity of these neurons with fire photometry during the fear memory retrieval test. And here's what the calcium responses look for all of those trials that he collected. And what you can see here by looking at the average response from these trials is that we didn't see a lot of modulation during the presentation of the tone, which leads to this fear memory retrieval. If anything, there's a bit of an increase at tone onset, which is somewhat consistent with this idea that PBT is important for fear conditioning. But June, he took this a step further, and he then classified trials because he noticed that in all of these trials were associated with different levels of freezing behavior. So he asked, well, what if we try to correlate the calcium responses from these different trials with the actual behavior of the animal. And this is what he did here. And you can see that here are the trials in which animals show high freezing behavior. Here's the trials in which they show moderate freezing, and here's the trials in which they show low freezing behavior. And lo and behold, what he observed indeed was that in the trials in which the animals display strong freezing behavior and moderate freezing behavior, those trials were associated with a reduction in calcium signal from these D2 positive neurons of the PBT, very consistent with what we saw in the active avoidance task, in which the neurons seem to be, as a population, signaling avoidance behavior, but their activity is being reduced during freezing behavior. So this was at least show that the activity of these neurons is consistent across both types of paradigms. And one interesting thing is to point out here is that this seeming correlation between the movement of the animal or the lack thereof during freezing behavior and calcium signal is something that emerged after fear conditioning, because we didn't notice any relationship between the movement of the animal and calcium signal in habituation prior to fear conditioning. Now, this is all was all very interesting to us, but it still remained a little bit of a puzzle, like I mentioned, based on prior literature on the role of PBT in fear conditioning. But we took a step back and try to think of this a bit further. And one thing that I wanted to highlight here is that we are looking at the activity of these neurons using fire photometry, which is a bulk kind of like bulk calcium imaging technique, where we're essentially collecting data from a large population of cells under the optical fiber. And as you recall previously, I mentioned that the vast majority of neurons in the posterior PBT are convex projecting. So it's possible that when we're doing calcium imaging, we are the signal that we're collecting with fire photometry is largely based on the activity or biased rather by the activity of these convex projecting cells of the PBT. So this is a question that we wanted to address directly, and to address this question, what June did was to, in this case, instead of using D2 Cree animals, June restricted the expression of GCAM to neurons of the PVT that projected to the nucleus accumbens by using a retrograde based approach. And essentially, to summarize the data that he collected, you can see here that consistent with the previous data from these D2 positive cells, we again observed that shuttling behavior is nicely associated with, or avoidance behavior is nicely associated with an increase in activity in these PVT2 accumbens cells, and that in general we see a reduction in the activity of these neurons during failure trials. Again, if we look at these task-related events, we see that like before, metrics like maximal velocity or the initiation of escape behavior were both associated with increased calcium chances in these neurons, whereas freezing behavior was associated with an attenuation of calcium in these cells. Interestingly, unlike the general D2-based imaging approach, in these PVT accumbens cells in particular, we noticed that seems like these neurons can discriminate a trial type, because the CS was largely associated with an increase in calcium signal in these PVT accumbens cells only in avoidance trials, but not in failure trials. So this is in contrast with what I showed with the D2 imaging approach. We also try to look at PVT projection to the central amygdala. So here, June imaged the axons from these PVT neurons over the central nucleus of the amygdala. And in contrast with what I showed you in the previous slide, we found that the CS in this case was more associated with activating these neurons during failure trials, which is interesting because these are the trials that animals are mostly freezing and not avoiding. And we didn't see any significant change in maximal velocity in calcium signals associated with maximal velocity. So this projection from the PVT to central amygdala seem to be a bit different than those that go to the nucleus accumbens. So we think that this data seems to be consistent with our previous hypothesis that perhaps when we're looking at general D2 expressing cells of the PVT, we have a mix of these two type of projectors of the PVT. So we decided to stick with this approach for the rest of our study to look at and manipulate specific pathways or outputs of the PVT. And this is what June did here. Specifically, in this experiment, he tried to assess whether these PVT projections to the nucleus accumbens indeed are required for active avoidance behavior. Having seen that this neurons seem to be sort of like a signal in avoidance behavior, he then wanted to use halorhodopsin to, like he did before, instead of silencing neurons directly in the PVT, silencing their terminals over the nucleus accumbens. And he took animals that were trained, and then he subjected them to these additional tests, one of which involved silencing of this pathway. And you can see here that, consistent with the data that I showed you before, silencing of this PVT accumbens pathway during presentation of the tone led to a very robust impairment of avoidance behavior that actually persisted a day later. And this is something that I'm going to get to back to in a second. But I also wanted to highlight that in trials in which the animals actually avoided, there was a marked increase in their latency to avoid, and similarly, there was an increase in freezing behavior. So it seems like these animals are kind of like shifting their behavioral response to the tone when this pathway is silenced. Now, this data was actually of particular interest to us because it was it reminded us of a previous publication by Fabrizio Domonte and Gregory Quirk, where in the context of Pavlovian fear conditioning and measuring freezing behavior, they found that silencing projections from the PVT to the central nucleus of the amygdala attenuated freezing behavior, and this impairment persisted a day later. In recalling their conclusions from this study, they concluded that these data suggested that this PVT projection to the central nucleus of the amygdala seem to be important for the maintenance of fear memory, because if you disrupt these projections during fear memory retrieval, you have a long-lasting impairment in their memory. Here, memory being assessed by how much time the animal spends freezing in response to the tone. And if you look at our data here now in the context of active avoidance and the projection from PVT to the nucleus accumbens, you see that it's actually look fairly similar, in which you get this impairment in avoidance behavior that persists the next day, right? And we were tempted to draw the same conclusion that perhaps this suggests that PVT projections to the nucleus accumbens are actually important for the maintenance of this fear memory or the behavior itself. Now, one particular observation that seemed to be in contrast or to contradict this initial conclusion is the fact that in these animals that have this lasting impairment in avoidance behavior, they also have a lasting increase in freezing behavior in response to the tone. So it doesn't seem like the animals are unable to remember the association between the tone and the aversive outcome or the shock. But what seems to be happening here is that they're shifting their overall response strategy to that particular association. So this led us to conclude, or to try to reevaluate the contribution of the PVT to fear memory, because both Domonte and Quirk and us, when I was a postdoc, made statements that the paraventricular nucleus of the thalamus seems to be important for fear memory, because if you disrupt it, you have these effects on memory, like maintenance, for example, as shown here, or as suggested by this data that I'm showing here. But we think that the data that we observe here suggests that the PVT's role in fear memory is really not in driving this CS-US association, but rather in tying this particular association to specific behavioral outcomes. So if there's something that, take-home message from my talk today, is that I think this lead us to think differently, or these findings allow us to think differently about the contribution of the PVT to fear related processes. So to summarize what I've shown you so far, I showed you that based on previous publications, PVT projection to the central amygdala seems to be critical for driving condition freezing behavior in fear condition animals. And today I've shown you that silencing PVT projection to the nucleus accumbens impair avoidance behavior, but also facilitate freezing behavior during the avoidance task. So naturally, a question that came out of this observation was whether when PVT projection to the central amygdala is silenced, which leads to impairment in freezing behavior, does this manipulation also impact avoidance behavior? Does it boost avoidance behavior? And this was actually indeed our prediction that it did. So to address this question, and actually let me take a step back and give a bit more context as to why this question has precedence. And that is based on a study that I mentioned earlier in my introduction from the laboratory of Joe Ledoux, where they were looking at active avoidance behavior in rats. And they found that, in general, a subpopulation of rats display this low avoidance response, where by the third training session, they were just not even breaking the 20% avoidance mark, all right? So they refer to this as low avoiders. And what they found is that these rats that were failing to show avoidance behavior actually display a strong freezing behavior. So they reason that perhaps the reason the animals are failing to avoid is because of their strong freezing behavior. And since they know this is a behavior that's central amygdala dependent, they did this lesion of the central nucleus of the amygdala, and they found that by lesion of the central amygdala, immediately they saw a jump in the rats' avoidance behavior. The next session they tested them, and this avoidance actually continued to improve through training. So this is consistent with the idea that, or suggests that the central amygdala is sort of like antagonizing avoidance behavior. So we wonder whether PVT, projection to the central amygdala, which we have shown to be important for these condition freezing responses, is particularly involved in this biasing of defensive behavior. So to address this question, June used again optogenetics to silence PVT projection to the central nucleus of the amygdala in animals trained in active avoidance. And to make a long story short, he observed that silencing this projection to the central amygdala improved avoidance behavior during this test session. And this was associated by a reduction in the latency to avoid, and also a reduction in freezing behavior. So again, this is consistent with prior literature that the central amygdala is important for freezing behavior, but it shows that this particular pathway is important for biasing the selection of these two types of defensive behaviors, because if you get rid of this particular output of the PVT, animals are able to engage in avoidance behavior much more. Again, June took this a step further, and he screened for mice that showed the same type of low avoider phenotype described by the Ledoux group. So specifically screen animals that after 3 training sessions display very low avoidance behavior. And then he silenced this projection from the PVT to central amygdala across additional test sessions. And what you can see here is that similar to what I showed before, and to the data from the Ledoux group, you can see that these animals improve their avoidance behavior with additional training when this particular pathway was shut down. And we did not observe such improvement in behavior in control animals, that instead of the halorhodopsin had a control fluorophore expressed in the PVT. So to summarize this part, I've shown you today that the PVT seems to be important for biasing the selection of defensive behaviors via its projection to the nucleus accumbens and the central nucleus of the amygdala. And if you silence one projection of the other, you bias behavior in favor of the opposite pathway. And you can see here, you can see this very nicely here in this bit challenging experiment that June did, where he expressed halorhodopsin in the PVT, and then he implanted optical fibers bilaterally, both above the nucleus accumbens and above the central nucleus of the amygdala. And he observed that when he silenced one pathway, in this case the nucleus accumbens, animals show impairment in avoidance behavior. But the next day, instead of showing this persistent impairment behavior that we've seen previously, if he silenced the central amygdala pathway the next day, these animals show recovery of their avoidance behavior. So show that manipulating these two outputs within the same subjects can dynamically affect the behavior. So with that, I'm going to summarize this part of my talk. Essentially, what I show you is that these D2 expressing cells of the PVT that signal active avoidance, particularly via their projection to the nucleus accumbens. Interestingly, these cells are also inhibited when the animal engages in freezing behavior, suggesting that there must be some kind of interaction across these two streams or outputs of the PVT. We're actually interested in addressing this question. I also showed that optogenetic silencing of this PVT projection to the accumbens not only attenuate avoidance behavior, but also drive these lasting impairments in avoidance and a concomitant lasting increase in freezing behavior, suggesting that the PVT seems to be important for switching the behavioral strategy to a particular CS-US association. And we saw kind of like the opposite effects when we attenuated PVT projection to the central amygdala. Now, I know I don't have much time left, and I definitely want to leave some time for questions, but I want to tell you very briefly about how we're looking to further these findings. One of the questions that we're trying to address in the lab is how these fear-related associations are transmitted to the PBT to support the expression of these different type of defensive behaviors. And we've done tracing studies in which we've used monosynaptic rabies method to map input from different regions of the brain, monosynaptic input onto projection-defined neurons of the PBT. That is, whether they're accumbens projecting or central amygdala projecting, we can now identify the specific inputs to these type of cells, and this is helping or informing our ongoing studies of how specific inputs to the paraventricular thalamus shape these defensive behaviors. And one input that we've been interested in, particularly in the context of this transmission of associative information to the PBT, has been to look in the middle prefrontal cortex. Largely, because in previous studies we found that the prefrontal cortex sends parallel projection to the paraventricular thalamus. In particular, based on our retrograde studies, we see that the dominant channel from the prefrontal cortex projecting to the posterior PBT is the prelimbic region. So, because it's the part of the PBT that we've been focusing on, we became interested in investigating the function of prelimbic projections to the paraventricular thalamus in the context of defensive behaviors. And again, there's precedent for this. The Fabrizio De Monte and Quirk study that I mentioned earlier also look at the contribution of the prelimbic cortex projection to the PBT in the context of fear conditioning. They found that this projection is actually required for the retrieval or the expression of conditioned freezing behavior. Similarly, in additional work from Gregory Quirk's group, they use this pretty cool platform-based avoidance task where the animal is like pressing for food here, rats, but they learned that during presentation of the tone, they can jump in this platform and avoid a shock. So this is like more of a conflict, a motivational conflict task, because the animals are pursuing reward and balancing their pursuit of reward with the threats in the environment. But what they found also is that silencing of the PL with mucimol also impairs the avoidance behavior in this task. And of course, more recently, a study from Cyril Harris' group, using optogenetics, which is more allows for more better, time precision in manipulating this area of the brain. They show that silencing of the prelimbic cortex, particularly during the presentation of the tone, as you can see here, also attenuated avoidance behavior in a shuttle box task, similar to the one that I show you today. So altogether, this is there's a lot of evidence that the prelimbic cortex, is important for the expression of the defensive behaviors, and based on its role in decision-making processes, we're interested in investigating whether PL projection to the PBT were actually important for driving avoidance behavior, and whether they may play a role in the biasing of defensive behaviors that I mentioned in the first part of my talk. So I'm not gonna, because of time, I'm not gonna get too much into this, but I just want to show you briefly some of our more recent fire photometry imaging data, where June used again fire photometry to monitor the activity of neurons within the prelimbic cortex that specifically innervate the paraventricular thalamus, here using retrograde methods. You can see that these animals learn avoidance very well, and that over time their freezing behavior or the amount of time that they spend freezing is attenuated. Unlike the previous study that we published, in this case, June wanted to see how this responses in this region of the brain evolve over time through training. So he recorded both during day 1, day 3, and day 5. And here's essentially the summary data for all the trials that he collected. And you can see the summary of that data here. And something that I like to point out, which was pretty interesting to us, is that with the course of training, you can see here day 1 is in black, day 3 in blue, and day 5 in red. You can see that during the avoidance trial, there is this increase in the responses of these PL to PVT neurons during avoidance trials that is not occurring in failure trials. You can see here that towards the end of training, there's no significant change in the activity of these neurons during the CS, actually during the whole duration of the CS. But here you can see that there's a persistent change or increase in activity of these PL neurons that project to the PVT, specifically for avoidance trials. So of course, this suggests that perhaps these PL to PVT neurons may discriminate trial type. So we look at a little bit more closely into this data using same type of analysis that I showed before, and you can see that indeed, if we look specifically at the onset of the tone, you can see that indeed there's this significant increase in activity in response to the tone by the end of training in avoidance trials that is not observed in failure trials. But if we look at a specific aspect of the behaviors like maximal velocity, we don't see much of a change towards the end of training, either during avoidance or failure trials, suggesting that perhaps this particular pathway is not important for the motivational aspects of the behavior, but it may be important for the more associative aspect that instruct the behavior, the decision-making aspect of the behavior. Interestingly, we did see that when the animals initiated their escape, this was associated with a reduction in activity of these PL projecting PVT neurons that again was clear for the avoidance behavior, but not for the escape behavior during failure. And in contrast, we see that the activity of these neurons is increasing towards the end of training to freezing. So it seems like these neurons do seem to perhaps be important for the behavior selection process, and that the behavior that the animal engages in is nicely tied with dynamics in this particular pathway. But this is something that we're exploring further, and we're trying to use other methods like single cell calcium imaging with miniscope, so that we can get a more rich data set and can try to address this question about dynamics within this particular pathway in the context of learning, but also in behavioral selection. Now, I think I'm out of time here, so I'm just gonna end my talk here, and I'll be happy to take any questions.
Thank you so much for that really lovely talk. It was everything, the data was beautiful. It was really very well explained throughout. We have a couple questions in the chat. And so I was going to say that if, let's see, Mike Schlund wanted to unmute himself and ask a question. I think he had a question that popped up during the first part of the talk with the PVT D2 GCamp imaging. Or if he's not available, then I am very happy to ask
I think I can see the question here. Is this the question about the percent of trials that are failure?
Yeah, so he asked about the percent of failure trials.
Yeah, so this actually varies. It depends on the level of learning in the animals. We try not to train animals for too many sessions because, with as you can see, with additional training, they can get to very high avoidance rates. So, we want to train them to a level in which we get like a nice mix of trial types, so that we can assess activity in this pathway for both type of trials. But people have, a lot of people have looked into specific aspects of the task, in terms of like the intertrial interval and duration of the tone that can actually drastically alter the level of avoidance performance in the animal. And I'd be happy to talk a little bit more about that. But in general, in our cases, we get mostly avoidance trial, but I think at least typically we get, 30% of the trials to be failure trials.
Okay, and then there's a comment from Jill Luttrell. Jill, you're welcome to unmute yourself, or if you're unable to, then I'm happy to read the comment for the audience. Okay, I will just read the comment for the audience, which is that this is very reminiscent of Kent Berridge's work, who's shown that the nucleus accumbens is an affective keyboard, excuse me. At one end, you can activate working for reward. At the other end, you can get defensive varying.
Yeah, no, that's an interesting point. I think we've actually looked at this same pathway, this PVT projection to the nucleus accumbens in the context of reward seeking, and we actually see pretty similar dynamics. Of course, we have not done an exhaustive, assessment of the diversity, projection diversity of neurons from the PVT that innervate the nucleus accumbens. We and others have found some interesting differences where there are classes of PVT cells that innervate more the core versus the shell, etc. But in looking at this pathway, with the type of manipulations that I've shown you today, we see that these projections are also signaling reward approach. So we think that this highlights a general role for these thalamus striatal projections in driving goal-oriented behavior, perhaps by engaging with motivational circuits. So it's kind of interesting to think further about how these pathways that this particular stream that seems to be important for both reward approach and goal-oriented defensive behaviors like avoidance may participate in biasing behavior amid motivational conflict, where there's the possibility of like searching for a reward when there's also threats in the environment. And I think there are multiple labs that are interested in addressing this question.
Wonderful. People can feel free to put questions in the chat, but I actually have a few that I would love to ask you about. The first is in the calcium imaging experiments, where you were looking at the calcium signal during the failure trials. I actually noticed, of course, like as soon as the tone finishes and I suppose a shock is delivered, that there's that large calcium transient during the period when the shock is on. And I was curious whether or not that large calcium transient is something that is always present during training, or it's something that emerges over the course of learning of like the active avoidance behavior, or whether or not you think that activation is driving any kind of plasticity.
Right. No, that is always present actually. So one of the things that I failed to point it out when I showed that particular data, but one thing that I had mentioned at the beginning is that we and others have shown that the posterior PVT, and in particular these D2 positive neurons, are particularly specifically activated by aversive stimuli. So in fact early studies on the paraventricular thalamus branded it as a stress sensor of the brain, because this region, particularly this posterior part, was or is strongly activated by negative stimuli, like shocks or restrained stress or tail suspension. So this is something that we always see, that an aversive stimulus leads to strong activation of these neurons. And that's why you see that strong response in the failure trials when they actually get the shock onset.
I see. And then I had one other question, which was, I'm fascinated by this result that you have of like kind of the lasting, toggling between the avoidance and the freezing behavior, even one day after the halorhodopsin stimulation. Um, and I'm curious whether or not, what the activity maybe during or maybe more importantly after halo stimulation looks like. I think that a lot of us who've used halorhodopsin see very high firing rates upon release from halorhodopsin stimulation. And so I was just curious if, it made me think like, oh, maybe perhaps release from that halo stimulation is maybe even activating those PVT neurons after the tone. And that could drive some kind of plasticity that supports the freezing over the avoidance behavior. I was just curious if you have any
That
thoughts.
is that is a fantastic question. We have not looked into that, but that's a real possibility actually, because of how these thalamic cells respond, right? And they have these strong IH current, hyperpolarization activated current, and they have these T-type calcium channels, both of which will lead to this type of sort of like post-inhibitory rebound phenotype. So it's a very interesting question, and we are trying to look at this from a different perspective, which is that we are so we recently identified actually that engagement of GABA receptors in these neurons is critical for the avoidance behavior, and it's kind of interesting to us because these neurons are actually activated during avoidance behavior, but it seems like at some point GABA receptor function is actually pretty important for driving the behavior. Actually, if we suppress GABA receptor function in the PVT, we see impairments in avoidance behavior in well-trained animals. But if we actually enhance postsynaptic GABA responses in these neurons, they actually have an increased avoidance responses in these animals. So, we have some preliminary data that suggests that perhaps there's inhibitory inputs to the PVT that are actually timing this behavior, meaning that they lead to suppression of the PVT and potentially a rebound of activity right after. So it's an interesting question that you posted. I mean, we have not looked at this. It's true that in the context of our experiments, the light is stopping right at the end of the CS, right? So one wonders whether that could be like reinforcing the freezing behavior or something like that. That's an interesting question. But although I cannot address that, we do think that perhaps this circuit is an activity. Activity within this circuit is largely shaped by inhibition, actually.
Okay, very interesting. I am sensitive at the time, and so I do want to thank you again for just a wonderful talk, kind of virtual round of applause.